So this is the stem rust data for the nursery in 2013. And this is the susceptible check, 1 or 2 genes, 2 or 3 genes, 4 or 5 genes, and then the single race-specific gene, all-stage gene. Without a fungicide, with a fungicide, and so on. And so what we hope and expect is that our fungicide treatment was effective. And if it's effective, we would expect to see a lower stem rust reading in the fungicide-treated plot than in the untreated plot. But it is higher here in date 1 of the first date the nursery was read. That's a little lower than in the second date. It was a little lower, so we're getting some protection. And on the final date, as the nursery is nearing maturity, we read it at that terminal rest score of a 40 ms for the susceptible check that was treated with the fungicide, and a 50 ms for the fungicide, the variety that was not treated with a fungicide. Did the same thing at another location in Ethiopia, and we had the same problem. On date 1, the untreated susceptible check read a 5 SMS and a 35 MSS, so this is higher than this. And on day 2, we read the same score, and so we really did not have the kind of rust protection we had planned for in the experiment. Some other varieties responded a little differently. Here's Franklin with 1 or 2 genes. Unprotected on day 1, it read a 30 MSS, but the treated plot read only a 5 MS. So we had some protection there. Protection seemed to continue all the way through the experiment at both locations. Danda with 2 or 3 genes, I think we also were effective with the fungicide, but we again did not achieve what we would hope for. This would be reading like a 0R or a 5R MR or something very low, indicated we had effective fungicide application. Kingbird, the fungicide application actually seemed to help, even though it has some very good adult plant resistance. And in the variety, the single gene variety, we also had some trouble with adequate fungicide protection. So those are cases where those comparisons tell us what I've just told you, that we really did not do a very good job of initiating our fungicide application. So we needed to initiate the application much earlier. We really needed more than 1 treatment. Tilt probably was not the best choice. We had some problems with stand establishment. This is hyena problems, hyena damage here. The birds ate some of it. So we didn't achieve the desired 0 effect. Good research starts with good agronomy. We had problems with seedbed preparation. We had stand establishment problems. We didn't have a problem with weed control, but we were wondering why we didn't have better stand, particularly in some of the whole units. And then we had virulence on SRTMP that showed up, so we had to change the experiment the next year, which is what we did. But we can look at some data. We measured yield loss for each of the treatments. So the variety PBW 343, for example, suffered a 33.8% reduction in 1,000 kernel weight. And this is despite having a very thorough, complete control of the fungicide-treated subunit. We didn't even have enough seed to measure hectoliter weight on this susceptible check. And yield would have been reduced by about 32.7%. For Franklin, we suffered a loss in kernel weight for each of the varieties, a loss of hectoliter weight, and a loss of yield. So these values are estimated are reported for 2 locations times 3 reps, so there are 6 observations going into that estimate, into each of those estimates and each of those estimates. So the example here is for Franklin, protected versus not protected. So the yield loss there, 658 grams. And you can calculate loss for one experimental unit, one comparison within a treatment. So these calculations here are for 1 rep at 1 location, but again, these treatments here are all based on a sample size of six observations. So we gave up on that data, pretty much. And we'll look at the data from 2014.